Next, I'd like to introduce Dr. Brian Berman, who came down here from VCU, where he's a professor emeritus, and he's has an endowed chair. Yeah, and he's the chief of Movement Disorders Division, and he's a chair of our BEBRF medical board, and we've served on that board together for several years. So he's also worked in Colorado, and he did a fellowship at the NIH. So please come up, Dr. Berman.
Thank you for the invitation, and good to see a number of you again. This time, the program has a different title. I was asked to talk a bit about imaging, functional imaging, that some of the research I do in its role in helping us figure out what's going on in blepharospasm. So that title is a little bit different. Just to get a little, continue the great talk that Dr. Dutton just did, just to talk a little bit about, functional imaging. So the brain activity is very active, metabolically active, and it brings in blood into the brain regions where you're active, and that blood changes the magnetic signal that we can detect. And so back in 1991, we realized you could measure this change in the magnetic state with oxygen, and that's called functional MRI. And this tool allows us to look at changes of the oxygenation or the activity in the brain, and then map it to what we think it should look like and determine if there are brain regions that are active or not active during a particular time or task. And what that's done is over the 30 plus years is in one way to find out, are there certain brain regions that seem to light up, as we say, or that are active during a particular condition, like the areas involving eyesight or the motor control. And a lot more recently, over the last 20 years, as I say, we look a lot about how the brain is connected. And so we see, are there brain regions that are metabolically active at the same time, and we assume that those are probably connected. So just reviewing some of what's already just discussed just a moment ago, blepharospasm clinically has this issue with the blinking can be increased, or there can be these abnormal closing spasms of the eye, and the eyelid movements themselves can flutter, or they may not open, as was discussed, and all these muscles that are nearby. And so when we study imaging, we're going to look at some of these features and try to figure out what's going on in the brain at the same time. And just a very high overview again about the pathophysiology. Currently we think that there's this hyperexcitable state that underlies the eyes and the eyelids spasming in the brain, or that comes from the brain. And we think this is a combination both of like some sort of predisposition state that maybe the brain is sort of has a loss of inhibition, or something that sort of creates ability to be overactive and hyperexcitable, and then environmental factors that seem to contribute with this baseline issue to create a state where the brain can actually have neuroplasticity changes. So, over time, these two things combine, and then you develop blepharospasm. So, why do we think neuroplasticity is involved? Well, as we mentioned, the average age is in the 50s, right? And maybe the exposures could have been some years before to some environmental factors, and so we know that there's changes over time occurring. But getting back to the hyperexcitable issue and the eyelid closing that's happening, you can think of this as almost like a reflex, just like when the pediatrician checks your knee reflex. We have the same kind of reflex in the eyes, and so the eyes have a reflex where if they're exposed to light or some wind or something that triggers a blink or closing of the eye. And so sometimes we use this reflex to sort of think about how what's going on in the circuit of the brain that could be going abnormal in blepharospasm. So I was thinking in the imaging, in a scanner, how could we elicit this sort of reflex in a scanner? And you could bring a big air. It would be hard to do a bright light in a scanner, but we could do air. And so ended up developing a study. And this is actual footage from someone lying in an MRI scanner where, so the person's in this fMRI scanner, and I have a tube with air pointed right at their eye, and then every time on a timer, blow air into their eye to make them blink and have this reflex. So we could do this as a way to sort of study what's going on in the brain during that blink reflex. So it's a fancy setup, lots of things going on here with cameras and air tubes and things like that. And then whoever is in the scanner is just looking at a display, and we have EMG, which is measures muscle activity, and then blow their air onto their eye to induce a reflex. And you can see here at the bottom that we have a timing when those blinks are occurring, but then there's also blinks that are occurring on their own, and so we had to go through all the time these long scans and look at it to make sure when they were blinking and when they were blinking due to the air being puffed. So just quickly, some findings. When you look at fMRI findings, you see these bright clusters here, and that's what we deem as being the brain activity of interest. And so in this case, the controls, so we did both controls and patients, about 15 of each. And in the controls, you could see that those puff blinks caused a reflex or caused activity in a part of the brain called the hippocampus, which is known to be functioning during conditioning, which is like getting used to things. So their eyes were essentially acclimating to the fact that they were being puffed. In the blepharospasm patients, actually, there was a high activity in the basal ganglia in a part of the brain called the caudate, and that has a role in regulating blinking. And so we saw overactivity in this region that we did not see in the controls. And then you can actually compare patients to controls. And what we found is that in the brain regions that are like the motor regions, so you would expect like a blink might be more forceful in someone with blepharospasm, that there was higher increase there, as well as in some areas in the eye cortex, the occipital cortex. And so this just sort of shows some brain regions that appear to be hyperactive in those with blepharospasm versus controls. So another thing that I did, besides causing people to blink in the scanner, is with the EMG here, is to just let people sit there for 10 minutes, and we watched had to ask them to keep their eyes open, but of course their blepharospasm is occurring, there's spasms, there's blinking, there's fluttering, and so we could see the activity of the blepharospasm in the scanner. Again, this is taken while someone's in the scanner. And so using this EMG information, we could then sort of do some fancy math and look at the motor activity, and then go back and say, during this motor activity, what's going on in the brain? And so if there's activity in the brain that matches with that motor activity of the spasm, that might give us some insight into that those are connected. You can also do some fancy math to look at the spasm just as it starts and just as it finishes. So you can look at the start of a spasm and then how bad the spasm was. And so this is something that we did in these 15 patients. And what we found is that at the onset, so the triggering of the spasm, this is called the putamen or the basal ganglia. And so we know the basal ganglia shows up a lot in this primary or isolated blepharospasm, and that showed up in the study where the basal ganglia appeared to be the thing that sort of triggers spasms. And then we found that if you looked at the severity of the spasms, that correlated actually with the motor cortex, which is what we'd expect, so there's more muscle activity, but also in the cerebellum. And the cerebellum sort of is an area that Dr. Dutton had raised too, that appears to be involved in blepharospasm. So I really like this study because it sort of showed these two aspects of spasms. One where like what's sort of triggering the spasm, and what is sort of continuing for it to persist as long as it does. And if we think of this more in like networks, as was mentioned before, these are kind of two different like motor networks in the brain. The basal ganglia one sort of seems to be triggering things, and this one in the cerebellum network and the motor cortex kind of is causing it to persist. And this is something that actually in other conditions like Parkinson's disease has been shown to be important for like tremors. So when people have a tremor in Parkinson's, it looks like the basal ganglia kind of trigger the tremor, and the cerebellar circuit sort of cause it to persist. So just something interesting to think about those two disorders. So this imaging is helping us understand the brain regions and the brain networks underlying blepharospasm, but still is what is going on, still hard to put together. So I'm doing another study where we can use MRI to look at the neurochemicals in the brain at the same time. And so looking at the brain chemicals that are involved in excitation or inhibition, it was brought up. These are the GABA and glutamate. And so looking in those same brain regions, the cerebellum, the motor cortex, and the basal ganglia, to try to see if we can see changes in the inhibition and excitatory signaling that maybe matches what we saw with the fMRI. And so just starting this so far with a different group of blepharospasm patients, but it does look like the amount of GABA, or the amount of brain chemical that's trying to quiet the brain down in the motor cortex, is correlated with the severity of the symptoms. So it's actually trying to, probably the brains are trying to, increasing the amount of inhibition because you continue to spasm. It's not enough to stop the spasms, but your brain is trying to suppress them. So with this is sort of is a introduction to the next sort of talks that you'll hear from other folks, is like, well, how can we intervene then? We know that there's this network dysfunction. There may be some signal that it's a loss of inhibition, and your brain's trying to, but it's not inhibiting it enough. And so one idea is that perhaps we can stimulate the cortex in a way that inhibits it even further. So, well, you'll hear some other discussion today about these techniques of using electrical stimulation or magnetic stimulation on the brain to try to change its state. It's either to excite it or to inhibit it. And so one thought is that since we know this network is trying to inhibit but needs further inhibition, that maybe we can use neurostimulation to do that. So that'll be talked about at another talk, and thank you for listening.